I find it quite challenging to give a talk, a remote talk, so really don't hesitate interrupting me.
Absolutely, we shall interrupt at will, and with a degree of politeness that would be quite revolutionary in economics, certainly. But I don't know about all other fields. Anyway, welcome along everyone today for our final leg talk of the semester. We have Silvia de Monte from the Eco-Evo Math IBENs in Paris, France, and also from Plön, where she's at the Max Planck Institute for Evolutionary Biology. So, yes, over to you, Silvia, please.
Thank you very much. Sorry, I was just figuring out. Do you see my pointer, or do I need to?
Yes, please.
Yeah. Okay. Good. All right. Thank you very much for inviting me. So what I'll talk about today is probably marginally connected to what you're doing, but hopefully you will find some interesting food for thought. My main interest is to study a model, mostly the ecology and evolutionary dynamics of collective behavior in mostly microbial populations. And today I will tell you about some recent work that I have been realizing mostly here at the Max Planck for Evolutionary Biology on how fluctuating fitness can influence diversity patterns in ecological communities. Let me start with some motivation of my interest for these communities. So microbial communities are extremely diverse and they are associated to very important functions, both in the biosphere, for health, for food production, whatever you can think of. There are behind biogeochemical cycles, there are a lot of different species of microbes that mediate the transfer of energy between inorganic and organic matter. And these communities, microbial communities, have been in the last 20 or 30 years the object of a particularly intense exploration since when high throughput genomic techniques became available. And a very interesting feature that I find very interesting in microbial communities is that you can use these methods to characterize the diversity of these communities in the wild, but you can also maintain to a certain extent diverse microbial communities in the lab, and therefore one can hope at least to do some experiments to test some theoretical predictions. Now, according to some estimates, there are about 10 to the 29 cells on Earth, and among all these cells there are millions of identified species, knowing that the concept of species can be tricky for microbes, let's say genomic clusters. And if you think of all this diversity, you can also imagine that the interactions between these different types also scale as the number of species to some power, probably. So we have this enormous complexity, and the problem that many people who have, like me, a background in physics ask is whether there is anything general to be said about all these communities. And in particular, we would like to understand something about the ecological processes that establish their functions as a community. Just to give you some ideas about what kind of patterns and processes I'm talking about, I will focus in particular on three features that are typical of every microbial community. First of all, microbial communities have a huge genetic diversity, and this is true everywhere. Sometimes, like in the gut of macroorganisms, this diversity is not enormous, but if you go out in the soil or in the water, whatever you use as a way of measuring diversity will result in coming up with many, many different species. These species do not have the same abundance, so what happens most of the time is that almost all taxa are rare, so they have small abundance, and there are only a few that dominate the community. And another feature that is especially associated to communities that live in the outer world and are not associated to hosts is that the species composition changes a lot in time and space. So let me tell you something a bit more precise about these 3 points. So how do we quantify diversity? These are just representations, schematic representation of what is called a rank abundance plot. So if you go in a given place and you measure how many individuals belong to every species, then you will come up with a number of species and the number of individuals for each of these species. And you can rank them from the most abundant to the least abundant. And then you will get a curve that looks a bit like this, that is called a rank abundance plot. And as you can intuit already, if this curve drops down very, very fast, then this would correspond to a situation of low diversity, because you will have a few species that are very abundant and then just not many that are rare, if rareness is defined as here in a very hand-wavy way as abundance below a certain threshold. But if you have high diversity communities, like typically those of microbes, what you will observe is that this tail will in general be much longer. So you will have many more rare species. A different way of seeing this feature is what is called the ubiquitous presence of very dominant species. So now if you look at what is the fraction of species that have an abundance that is 50% of the total, you will see that when you have communities that have more and more species, here in green you basically find the bacterial communities that have a huge amount of species, until thousands or tens of thousands for a single sample. You will see that the abundance will be more and more concentrated in the sum of all the rare species, and then there will be few very, very abundant species.
Okay, hang on, that's good. Can you just go back? Okay, so this means that if we take, say, 100 as the number of species, this is if we're there, and then we've got the fraction of those species that comprise 50% of the total in measured by, units of organism, right?
Yes, just number of individuals, considering that these studies are made by taking individuals that are of the same kind, so you don't mix bacteria with trees.
I
so
see.
you take trees and then you just look at what is the distribution of individuals in among the species of that tree.
So the further right you get, the less diverse, according to your previous setup?
So no, it's not less diverse. It's just that the mass that is in the tail is higher.
Right.
So you have the species that are very dominant, they are actually fewer. Okay. So when you add number of species, you are basically adding to the tail of rare species.
Okay, thank you.
And you have a few very dominant ones. So just another way to see this that is associated to especially these patterns of dominance in rare communities. What you will typically observe is that dominant species do not seem to be dominant forever. So there are some species in certain circumstances that are always relatively abundant, but very often you have this kind of very intermittent dynamics where you have that a species becomes dominant for a short time scale, and then it goes again back in the rare species, and then you have another one that becomes dominant. So I'll go back to this kind of data and the way we interpret this, but this turnover dynamics makes so that basically the dominant species not only are a few, but they are not always the same necessarily. Okay, so in order to quantify this kind of observations, people use basically the most complete kind of representation you can give to the data you have. I remind you the data we deal with are the number of individuals that belong to different species. So this is, we can, in order to know more of this, we really need to enter the functional ecology and to go and see exactly what species is doing what. But if we don't do this, this is the information we have. And when people talk about measuring diversity, for instance, they are just making statistics over this kind of data. So what people usually do is that they take directly the distribution or the histograms, if you want, of this data that are called the abundance distributions. And these distributions, as I will show you, there are different kinds. You can make, you can look at what is the histogram of the species that are present in a given community at a given time, or if you have a time series, you can look at the frequencies of occurrence of a given species over time. And the good thing of these distributions is that they can be compared to predictions from models. And the hope has been for a long time to be able to tell apart what model is best to describe the distributions that are empirically observed. And this debate has been going on for years and years. There are plenty of different models that predict different kind of families of functions, and then people try to fit this to experimental data. Just to give you an example, so imagine that this histogram is derived from experimental data. This is actually a simulation, but it's very similar to what you would obtain for a typical tree community. So this representation is a bit different with respect to the rank abundance plot I showed before, because this time you're counting how many species have an abundance that is contained in a given interval. And this is usually, people do this in order to have more regular kind of plots that are easier to compare to models. So this is typically, I think this is in power of 2, sometimes it's in power of 10, but so you get this kind of distribution. And then once you have the histogram, you can try and fit it with different classes of distributions. And this is where the whole debate went on. Perhaps this model is giving me something that looks a bit more like the empirical observations. Now, just to give you one precise example, this is a work that we did on plankton communities, so plankton protists, unicellular eukaryotes that live in the open ocean. And we had about 400 different samples of different size classes, different locations in the world ocean. So sometimes very, very different biomes. And what we did was to plot sample by sample these species abundance distributions. And then we fitted all of them with one law that was derived by a model that's, let's say, in the class of neutral models for those of you who know what they are. It's a bit like Hubble's model, but with density-dependent birth and death rate. So this model allows to evaluate what is the dominant trend. In this case, it's a power law trend in this sample. And also you can exclude, it gives you a criterion to exclude the very abundant species that are typically distributed in a way that is different from the rare species. And by doing this, a bit to our initial disappointment, we realized that everywhere in the world ocean, rare species follow the same distribution, not only in the sense that they are all distributed as a power law, but quantitatively, the slope of this power law has a variation that is less than 8%, which is something that was really surprising to us because certain data come from regions of very high biomass and others from ocean deserts. So this, basically the same law, or better, let's say the limit of this distribution was used recently and applied to several thousands of different samples. So Gao and colleagues found that this kind of distribution, that is the product of a power law and of an exponential distribution, seems the best to fit the species abundance distributions in all these samples once sampling is taken into account. And sampling means that, when you have less resolution, if you have less individuals that you are sampling, then you will have some noise in these distributions. So they didn't look very much quantitatively into the difference between samples, but for microbes, they typically find exponents that are in the range, let's say, between 1.2 and 1.8. So more or less the range that we also observe for phytoplankton. At the same time, other people have worked on similar data, and they found laws that look a bit different. And they look more like the kind of distributions that are typically found in trees and in macroorganisms. So these distributions are characterized by the existence of like a peak of in the SAD, and then the theoretical prediction would be that the abundance then decreases. So this was basically the starting point of our work. We were wondering how is it that certain people publish papers saying that this law is actually universal, it works everywhere, and somebody else publishes a different law saying it works in every case. We wanted to understand how is this possible, and when should we expect these laws to be the same or different. So this led me to collaborate with Emil Malming, a brilliant postdoc here at MPI, who has spearheaded this project in collaboration also with Arne Trausen. So what we wanted to understand is what are the key features that really matter in these species abundance distributions, and we also wanted to understand what ecological factors are shaping these features. In order to do this, we started with a very simple model that is really super simplistic, but that sort of contains the basic hypotheses that are found in all other models for community dynamics and community ecology. So this model is describing the variation in time of the abundance of species I, and this is composed of a per capita net growth rate, that is this term, that contains three parts. The first part is, if you want, an unconstrained growth rate, or I'll call it fitness. Some people would not agree with calling this fitness. Anyway, let's say it's something that can change over time, but in a way that is independent of the other species. or actually of all other individuals in the community. And we assume that this, and perhaps it will be more clear later why this is the assumption we take, but we assume that these growth rates are fluctuating over time. They have an average, so every species has an average fitness, but at every time, every species has a different fitness, and this fitness fluctuates according to a given time scale. And this is so. This is basically colored noise. So it's a color noise fluctuation of the fitnesses. And then we have two other terms that model the interaction with other individuals of the community. So the first term is the same for basically every individual. There is like a density dependence on the total number of individuals in the community, whatever their belonging is. So it doesn't matter what species they belong to. And then there is an additional self-limitation term, and this is specific to the individuals of the same species. So you can compete with all the other individuals of all the community, and here we do not distinguish among species, or you can compete with the individuals of your own species, and typically this epsilon is considered in ecology to be positive, in the sense that you interact, you compete more with individuals that are identical to you than with individuals who are different. And this is just because you are sharing the same niche, and therefore it is expected that competition is harsher within a species than between species. Finally, we have this immigration term. You can think of it as immigration because these are like local communities, but you could also think of it as speciation, so something that basically prevents a species to be permanently extinct or exit from dormancy. So there are different interpretations one can give to this term. Now, in order to, oh, sorry, I forgot. This parameter, gamma, is very important. I will show you immediately after that this is related to the exclusion timescale in the community. And this depends on the timescale of the fitness fluctuations and of their variance. So just remember that when gamma is larger, then you have, faster and stronger fluctuations in the fitness. So let's start from a simple case that we call the neutral case, where it's neutral in the sense that every species has the same average growth rate and has the same density dependence. Okay, so basically it's a species symmetric kind of model. And now if you look at the difference between the instantaneous fitness and the average fitness, every species has basically the same kind of trajectory. Just at every time it will be a different species that has the maximum growth rate. So now some people recently have claimed that this is enough to explain why a lot of different species coexist. And indeed, if you make a simulation, what you see is that there are species that seem to dominate, and then they basically go close to extinction, and then others will dominate. So they call this the stickiness of rarity. So the dynamics slow down when you become rare, and therefore it takes a long time before you can exit this state of rareness, and this gives the opportunity to other species to become dominant.
Right, hang on, but this is just this is driven by the randomness in fitness at any given time, right?
Yeah, this is unpredictable. So sometimes some species happens to grow faster than the others, and then they will grow for a while. Sorry, I forgot to tell you about this. So if you start with a distribution that is peaked so that all species have the same abundance, this will lead to the distribution of abundances to become larger and larger. So here you have the different colors are just different times over. You just look at the snapshot abundance distribution. So you take at a given time the distribution of all the abundances of all the species, and what you observe is that if you start with something that is very concentrated, it will become larger and larger. So basically, in the end, you will have a lot of diversity that is due to this, just a stretching out of the abundances. The problem is that if you don't have...
Sorry, can I also ask a question?
Yeah, sure.
So you have, there are two terms. One is the fitness term and the other is kind of limitation term with a parameter mu. So that is also important, the limitation term, to get this picture. If you have mu equals 0, then you have explosive growth of some, I imagine.
So mu is basically the carrying capacity. So it determines what is this upper boundary of the abundances. So these are abundances. The lower boundary, so there is no lower boundary in this case. Species can go as close to 0 as they want, but they cannot become larger than 1. So typically, so this fixed parameter that is not 0, indeed. So if it were 0, this would diverge. So and then it would grow forever. So the fact is that it doesn't actually stay like this, because if one waits long enough, actually there is one species that becomes dominant and all the others basically disappear. And if you fix a threshold below which you have extinction, then they will all go extinct except one. And if this threshold is, these are the example of 4 different thresholds. So as you reduce the threshold, it takes a longer time to get to fixation. And if you don't fix any threshold, you will have all the time this sort of fact that rare events will make the dominant species change. But then it will always be one that is very dominant and all the others would be basically 0. So there is exclusion, competitive exclusion, that occurs on a time scale that scales like the logarithm of S is the number of species over gamma. And this is why 1 over gamma is what I call this the time scale of competitive exclusion. So this can be analytically demonstrated because if you turn, so if you go from this like Lotka-Volterra-like formulation to a replicator equation, so you are describing the frequency of species I in the population, then the formal solution of the equations of the replicator equation looks like a Boltzmann distribution. And in this distribution, what actually matters is the integrated fitness over time, whereas the instantaneous fitness is different from every species, but on average it is the same. This time integrated fitness is instead a diverging quantity. So there will be in the end one species that is actually dominant over all the others. And this is associated exactly with this time scale that, so this can be mapped in the random energy model in physics, and the transition from a case where you have many species to monodominance is basically like a condensation in physics. So you really have a sort of a phase transition between a very diverse community and a community that is dominated by just one species. So it is not enough to have fluctuating growth rates to explain why you have the coexistence of many, many different species. So what can one do then? This is the picture I showed before. So basically, if you keep going in time, this will end up having just one peak around 0 and one peak around the carrying capacity. But now you can start adding self-limitation, so the fact that intraspecific competition is stronger than the competition with other species. And one can also add immigration. Now, if you add these two features, what happens? Yeah.
Yeah, the new one. Yes, I mean, but this is not one, but the peak and
Excuse me, I didn't hear you.
Is that a question from Luciano? Have a question, or is this a mic issue? I think it might be a microphone issue. Okay. Please continue.
Right. So basically, the moment in which you have that these communities are put like in a meta-community context, and when there is self-interaction, then you don't have anymore this problem of extinctions, because you have immigration, and the existence of this self-limitation makes so that what we observed previously as a transient gets stabilized, and this becomes like the asymptotic dynamics is basically the transient that we observed previously. Now, in order to make this a bit more precise, let's consider again a simplified version of these meta-community models. So we again take that every species has the same average fitness in time, over long time. And then we consider that immigration is the same for every species, and self-suppression, or if you want, intra-specific competition is the same for every species. Okay, and now what we can see is that with these features, the time, the exclusion time scale basically diverges, and we can maintain this out of equilibrium dynamics virtually forever. Now, it looks a bit different from what we had before, because you don't have that the dynamics slows down, but it keeps changing over time in something that looks very much like what is observed in data. So here are two examples. This is an example, the first one from some metabarcoding of bacteria communities. So people measured genetic clusters in the bacteria communities every day for, I forgot how long, but a few tenths of days. So they could see that the dominant clusters would change massively over time, and on time scales that are much faster than the time scale of environmental variation, that is typically associated to seasons. This is like a coastal station, so you don't have huge turmoils of transport due to transport. And similarly, very intermittent kind of dynamics was observed this time in a lab experiment where people took a plankton community and kept it for 3 years in the lab, and they could see that dominance patterns were very changing. So this looks a bit like the kind of simulations I showed you before, but it looks also perhaps chaotic. So there is a bit of a debate in this moment whether these fluctuations are due to some kind of deterministic chaos or they are due to stochasticity of some kind. They are not seen just in these two cases. Is a nice work by Tanya Rogers in the group of Steve Munch. They found out by analyzing a huge amount of time series that many time series show hallmarks of chaos, and this is particularly true for plankton populations or communities. Which kind of chaos this might be? Well, this is difficult to say, but let's say long ago people proposed simple models that had chaotic dynamics. And this chaotic dynamics was due to some pretty nonlinear choices for the interaction functions. And therefore, this has not been really completely believed by people in the field, because they said, okay, this happens only for a very specific choice of the parameters, so it's not robust enough. Now, it turns out that in the moment in which you have many, many different species, chaotic intermittent dynamics becomes like a generic feature that you have in a broad region of the parameter space. And therefore, this might be the reason why you see this kind of apparently chaotic intermittent behavior. However, even in this case, so in this case where you have a very high number of different species, every species feels the effect of all other species in a way that resembles or is basically identical to that of having stochastic fluctuations in the environment. So it's very difficult to tell apart, what is deterministic chaos and what is stochastic fluctuations in the environment. So I will leave apart this discussion on chaos and go back to the stochastic fluctuations. Okay, now I'll go back to the model and just give you some elements on how one can use the model to understand what's going on in the patterns that are observed, empirically observed. So a first observation is that if you take these equations up, you can rescale them. And once this rescaling is done, it turns out that there are only two parameters that are really important to determine the nature of the distribution of abundance. And these parameters are the ratio, the log ratios between couples of these three parameters that are related to self-limitation. This one is related to what we call buffering, so basically immigration, what avoids extinction. And the third one is related to the time scale of competitive exclusion. Now, once you have these ratios of these two parameters, you can project any of these systems that depend in general on 7 different parameters on a space that is just a plane. And in this plane, you can recognize regions where buffering is dominant or self-limitation is dominant, or even see the effect of changes in the time scale of exclusion that lets you move along the diagonal. So just to give you an idea, I will show you 4 examples of simulations that have been realized by fixing these parameters, so fixing just these 2 compound parameters, and then choosing all the other parameters randomly so that these 2 parameters remain the same. Do you follow?
What's the little extra the set of axes in the left bottom corner of that?
So if you increase self-limitation, then you're moving in the vertical.
Yes, that's right.
Increase buffering, so if you increase immigration, you move in the horizontal. And if you increase the time scale of exclusion, you are moving along the diagonal towards the origin. Towards the origin.
Okay, yeah, that's clear. Thank you.
So and now let's consider, for instance, this point. Okay, we fix this compound parameters, but then we make a lot of simulations where we vary of orders of magnitudes the other parameters. And now the expectation is that if this rescaling makes sense, it should not matter what is exactly the value, the exact values of all the other parameters. A second observation is that we can try and describe the behavior of typical species in the community. Since these models, so far we have assumed that the growth rates are statistically equivalent, so the fitness fluctuations are the same for every species, we can think that actually we can swap the species and get a description that holds for the typical species. So we choose an effective model that has the following form. Is basically a logistic equation where the effective growth rate can be chosen according to what a given trajectory or simulation provides. And similarly for the variance of these fluctuations, and the effective time scale is basically dominated by the time scale of the fitness fluctuations. This model has the advantage that can be solved, at least in the limit where tau is small, but this approximation, as you will see, holds basically for every tau. And this provides an analytic solution that has this form and that allows to have to directly connect the exponent of the power law distribution to these effective parameters. And what is interesting is that the shape of this distribution is what is called the generalized inverse Gaussian distribution. It depends basically on 3 parameters, and then we can use these 3 parameters to fit the observational data in principle. Now, before going to the data, I mean, we haven't really played a lot with the data so far, but I will show you what is the results if we run a lot of simulations with a lot of different parameters, as I was mentioning, chosen from orders of magnitude of difference. So what I will concentrate on is the shape of the abundance distributions. So just as a reminder, when you fix time, you will have species abundance distribution that is basically the blue diagram, the blue histogram. So this will tell you how many species have the abundance in a given interval. But then you can also take one single species and look at what is the distribution of the abundances in the course of time. And this will give you like the pink dots. Now, whenever species have the same average growth rate, these 2 distributions coincide. So taking one or the other is the same. And then one can quantify the shape of this distribution by looking at 3 main parameters. First of all, the support of the distribution, the extent, basically the range of abundances. Then you can take the peak of the abundance, like the mode of this distribution, and you can take, which I will call n star, and you can take the slope of the power law part of the distribution. Oh, sorry, I forgot. What's going on? Okay, sorry. So if you remember the 4 points I showed before, this is what happens is that for 1 point you get, by changing all the parameters, you tend to get distributions that have more or less the same class of shapes. And this class of shapes, they actually span all the kind of shapes that have been observed in experimental data, sorry, not experimental, in observational data. So depending on these 3 compound parameters, you would get something that has very long power law distributions or shorter power laws, or you may have something that looks basically like a log normal, or like a gamma distribution, so a distribution that is skewed towards the left. Indeed, what you have is that as you move in this space, you are spanning the whole continuum of these shapes. So you don't have just one shape that is the best for everything. It depends on what regime you find yourself into. Knowing that these different regimes, they are quantitatively different, but there's no like a qualitative cutoff that separates one regime from the other. And now this is a different way to show that basically most of the shape of the distribution is controlled by just these two parameters, so the buffering and stabilization parameter. As you see, when one makes a lot of different simulations, randomly choosing parameters, the value of this different shape parameters is basically a gradient, has a gradient in this space. And here you have that part of the space is in gray, because these are the regions where you can't really fit a power law in a way that is reliable. So we have obscured this. Okay, there is a geometric intuition, but I'm running late, so if you want, we can discuss about this later. I'll just get back to this, apparent dichotomy in the data. And now we sort of start understanding why you have this very different kind of shapes. Actually, this is typical of data that come from very deep sequencing, where rare species are really well characterized. However, this data, they come from a public repository where rare species are basically discarded because only species that have a certain occupancy are present, as people want to avoid taking into account something that comes out of errors of sequencing. So somehow these data are more reliable for estimating the species abundance distributions of abundant species, and these data are more reliable for estimating rare species abundance distribution. And now, as in any natural system, not all species will have exactly the same average fitness. Then we looked at what happens if you spread out these average fitnesses. So this corresponds a bit to considering that some species will be more dominant just because their average fitness is higher, and some other species will be more rare because their average fitness is lower. And now what happens, these are just two examples of my four different cases. What happens is that as you spread this average fitness, which can be also very, very different, here we have the difference between so the difference in fitness is 10 times the sigma, the heterogeneity in the growth rate that is given by the noise. So what happens is that some species, the species that happen to be the fittest, they will tend to have distributions that look more like those that are, unimodal or left skewed, whereas the species that are more rare, they will rather have something that looks like a power law. And if you integrate over all the species, you will get a distribution that looks very much like those that are observed in the environment, with a long power law that is only revealed if you have a sufficient number of rare species. Otherwise, you will basically have gamma or log normal distributions. The coexistence of different distributions depending on what species are actually well fit to the environment and which species are instead less fit and they come visiting this environment every now and then was actually previously noticed in fish populations in this very nice work where, since people could actually know what was a given species, something that I mean over time, something we cannot do with genetic data, then they really saw that the species abundance distribution is actually the superposition of different abundance distributions that are different between abundant species and species that are occasional. And finally, the model also allows to look at something that has to do with turnover. So experimental data show that if you look at how much a given community changes over time, you will have a decay in similarity, but this similarity doesn't seem really to go to zero. So this is something that is observed in the model mostly when you are on the, let's say, top right part of the diagram. So when you have species that are very abundant and they have a unimodal distribution, in that case, actually, if you increase heterogeneity, you will get that basically dissimilarity decreases very slow. If you instead want to explain this sort of intermediate decrease of similarity, that is intermediate turnover, then the best explanation is that there is this very chaotic boom and bust dynamics, but there is at the same time heterogeneity in the average fitness. So I come to the conclusions. So the combination of fitness fluctuations that yield the growing inequalities in integrated fitness, of buffering and of self-limitation constrain the abundance distributions in a way that is very generic. Basically, your distribution needs to exist between two boundaries, and it only has a small, or if you want, a continuous number of different shapes. So the statistical patterns that are observed empirically are retrieved by this model, and all that one has to do is to change quantitatively some parameters that are unfortunately the aggregate of many microscopic parameters. This means that it is impossible, in principle, to derive from these distributions, even the time-resolved ones, what is the underlying process that creates them. So this is a sobering message, but at the same time, not all distributions are the same, and we can hope to exploit the qualitative shape, or at least more or less how the quantitative values of the fits to these distributions to be able to understand what regime do they belong to, and to link this, for instance, with temporal turnover and other kind of ecological patterns. And this is all. Thank you very much for your attention.
Thank you very much, Silvia. I shall stop the recording and we can ask some questions.